about inductive. Now I just started the recording, so let's try that. I understand if you're having troubles with inductive and deductive reasoning. Like I said, I even do it myself. Yesterday we made the all living things are made up of cells, and we talked about that, and we said it was inductive, didn't we? I think we did. I'm pretty sure we did, because it's not. That's because half of the sentence is missing, and this is where it gets confused. You remember inductive and deductive reasoning, and this is why I wanted to go over this, because you will have some questions about this on the exam. We're going to talk about that later, but I am going to have some examples of statements, and you're going to have to tell me, just like we did in class, so practice those. But remember that figuring out which is which, it's the way the logic flows, all right? So this is why that sentence is only half there. All living things are made up of cells. So something I'm looking at that has cells is a living thing. All right, so we go from really, really big to really, really small. That's why that's deductive reasoning, not inductive. I'm sorry, I confused you. Like I said, it's because only half the sentence was there. When you start with something really big, look at the third statement. All cells come from pre-existing cells through cell division. The other part of that sentence then would be that if I see something that is a cell, it must have come from another cell. I make a prediction. So deductive reasoning. When you go from big to small, when you go from really everything to me, my roommate, this cell, then you're deducing, you're removing, you're making the logic kind of get more narrower. Whew. Does that make sense? I'm sorry if I confused you yesterday, and if I said you were wrong in class. I think we went right to inductive. I'm like, oh yeah. New blood from old. Give me a little more. I assume you mean a garden implement. Like if you're making, you mean that occurs in your bones, or okay, yeah, your bones produce red blood cells, the marrow of your bones. Is that what you mean?
No, when you get your blood taken and they
Yeah.
want to see your blood levels, they can tell you if you're making new blood or old blood. And if you're not making new blood, then that's a dysfunction. But because the cells are tiny, they can say, oh, those are the old ones, and you are making new ones, and the small ones are the new ones.
Okay, I'm going to take they're not very big. Red blood cells are 5 to 8 microns, so they're really, really small to begin with. So I doubt it's a size thing. Old blood cells, remember the hemoglobin protein? It's very, very stretchy, like we talked about. The reason why that protein is so high in red blood cells is red blood cells have to often squeeze through areas smaller than they are. Capillaries can get down to 1 millimeter in diameter. And I told you red blood cells are 5 to 8. The way they do it, I wish I had my pen, it's working, but they squeeze through. They basically elongate. As red blood cells get older, they don't elongate as well. They're not as springy, and they tend to fragment and come apart. That's not good for something that's trying to squeeze through a small opening, right? All them little chunks plugging it up. So in your spleen, believe it or not, there's a thing called, if I remember right from my A&P days, a squeeze test, where red blood cells are sent through a small little opening to see if they'll fit through an opening smaller than themselves and not come apart. And if they do, they actually become part of your bile, right, which is dumped out into your intestines eventually. So back to your original question, maybe how they know old from new blood is checking the stretchiness. Like they can somehow measure the elasticity of the cells, and they know the less elastic a cell is, perhaps, or the older it is, right? I don't know for sure, but I'm just taking a guess. I don't know. I think it'd be hard to see a newborn red blood cell. They'd be really, really small. Okay, so good question. Cool. Anybody have any other questions for me involving the inductive and deductive reasoning? As I said, please take a look at that sample and that we talked about in class. Look at them online. Type into Google inductive versus deductive. and you'll find columns of one say, this is inductive, this is deductive. And do that test. Does it go big to small or small to big? Do it like riding a bike, right? Like riding a bike over and over and over and over and over again. It becomes repetitious, which I don't understand why scientists always seem to have troubles with it. Who knows? Probably because the words are opposite when you look at it. All righty, on to cells. So we started off with very simple cells, right? We looked at prokaryotic cells. Not trying to make you seasick here. Prokaryotic cells, relatively simple critters. Hey, someone stop me at, let's make it 8:45, okay? And then I'll start doing the review. Don't let me keep rattling on. All right, here we go. So prokaryotic cells, relatively simple, although that's not the best word for them, right? But right, not a lot of complexity to them, perhaps. One chromosome, we have a bunch. Some of us have more, some of us have less, but they just got one. It's all crammed in there, all folded in on itself because it's really a loop. So you fold it, fold it, fold it. Now don't confuse that with proteins folding. This is something different. This is a nucleic acid, right? Chromosomes are nucleic acids. That's your DNA and RNA. But it's folded up and it's not shoved into anything like in our cells, like in a nucleus, but it's shoved into a region, right? A nucleus, oh, no, two different chapters. I could have made a cool test question. A nucleoid in a prokaryotic cell is analogous to what part of the of atomic structure? So a nucleoid, which is not really there, it's just an area, is similar to some structure we talked about that atoms have. Yeah, that's a good question. I like that. Unless you're all going, what in the heck is he talking about? Remember we talked about atoms and we said, okay, don't confuse. There's terms that we use for atomic structures. One of them really exists, and the other one is a grouping that we made. Come on, I'm doing a better job of hinting than that. Hi, Wyatt. How are you today? Ah, thank you. Yes, it's a shell, right? Shells don't really exist. Good. Thank you. Orbitals exist, right? That's where electrons are. It's a collection of orbitals that is a shell. All right, so that's the same thing as the nucleoid. If somehow the chromosome of the bacteria are crammed up on one end, that would be the nucleoid, or the nucleoid region, sometimes it's called. And if the chromosome were at the other end, well, there would be the nucleoid. But when you get to your typical animal and plant cells, and I'm type this word in, we move from talking about prokaryotic cells to eukaryotic cells. And I told you in class yesterday what a kary was. Yeah, remember that's a nucleus. And then somebody said in class what U was. Y'all remember what the what do you call that thing? Tidbit. Oh my goodness. The prefix U means when you see it. I'm trying to think of another word. Well, this is an awful word, but it is a word with EU at the beginning. It means the same thing. What does EU mean? Don't confuse that with emu, which is a bird. You know how I remember? No. How I remember it? Goo? No. Okay. Yeah, Raymond, let's kind of run with that. Good, true, exists, is there, or something like that. Remember, pro from yesterday for prokaryotic meant before. Like, oh yeah, like when airplane, Boeing and all them folks, when they're going to make a new type of airplane, they make a prototype first. Okay, they make a model. So the prokaryote is before the nucleus because it ain't got one, and then the eukaryotic is organisms that have the nucleus. There it is, right there, right? Big old nucleus, big old an actual structural there. I always remember EU is true because the word true actually has the letters, but they're reversed. But whatever, I'm a very A to B kind of remember. Like, oh, okay, when you see this, think that, right? Yeah, absolutely. Okay, so we're going to talk about all these parts, but this is a picture that we call a composite picture. And I don't know if you've ever seen that before or heard that, but what that means is not all cells have all these parts, okay? This is basically a cell with all the parts, and some cells may not have these parts based on what those cells do. And some cells might have more of these parts, and some cells might have fewer of these parts. Like, back in the red blood cells, interesting enough about red blood cells. Red blood cells are cells, and I'm going to hopefully not confuse you, but they don't have a nucleus. They're anuclear cells. Basically, what a red blood cell is created for is to carry blood. So they kind of strip it of all its things except hemoglobin. Even restricts it down to getting rid of a nucleus. Let's just make it bare bones. That's why at crime scenes, when they get DNA off blood, they don't do it off the red blood. There's no nuclei in red blood. There's no chromosomes. They do it off the white blood cells, the immune cells that are in the blood, right? Because your immune cells do have a nucleus. Now when you look at, what is it? What are those things called? Those forensic shows. I don't remember. 3 letters. NIS? Whatever it was. No, that's the other one. CSI. Yeah, there you go. All right, so we'll talk about each of these parts in more detail, but as you can see right away, when you look at this, one of the first things that stands out, right, is the fact that, wow, there's a lot more going on there than prokaryotic cells. There's a lot more going on there than bacteria. And there is. Again, let's not slight the bacteria. For one thing, you don't want to tick them off. There's a lot more of them than us. But they do, as my professor used to say, they do so well with so little, don't they? What I want to do in the next 2 slides, this one and the one next over, is I kind of want to move back and forth. I want to do that compare and contrast thing I like to talk about. So don't focus on the parts so much, all right? I mean, I'm going to be talking about some of the parts, but we're going to go through each one of these in detail later on. This is a typical, or a composite, if you want to use that word that we used, an animal cell. This is an animal cell. I can look at this right away and typically tell that this is an animal cell. And I'll show you what I mean, because there are basically 2 types of cells out there. Fungi a little bit different. Okay, don't worry about them. But anyway, there's 2 types of cells out there. There's there's more than 2 types. There's 2 big types. There's the animal cells and their buddies, the plant cells. So this is your composite or typical plant cell. Right? And if we were comparing, right away we can see, well, let's see, there's a nucleus. Yep, got one there. Let's see, we got this endoplasmic reticulum. There's some of that right there. It's ER. If you said, what the heck did you just say? I said endoplasmic reticulum. That's such a neat word. There it is. I like these kinds of words. Endo is within, right? Like if you have an endoparasite, you have a parasite that lives inside of you. Plasmic, the liquid part of the cell. Can I label it here? No, they're not. But all these things that we, yesterday in class we called organelles. The nucleus is an organelle. The endoplasmic reticulum is an organelle. It's a play on this word, right? Organs, which is what you're made of. Not a play, a variant. But all these organelles are floating in this sort of semi-gooey material known as the cytoplasm. Okay? All in between all those organelles, that's what they're floating. And not floating in air. They're floating in this sort of, like the gelatin yesterday a little bit. Right? Remember that gelatin stuff that we messed with in lab? Maybe not quite that thick. But they're kind of floating in that. And this organelle, the endoplasmic reticulum, what it does is within the cytoplasm, right, endoplasmic, it reticulates. Reticulates. That is a word. That is an act. That's not a, well, it is a science word, but it's a word that you can refer to lots of things. If you were walking down the road and saw a snake, you might say that a snake moves by reticulating. If you were some, nerd scientist, right? So here's that snake moving down the road. What does it mean to reticulate? If you weren't very good at walking on a pitching ship, there you go. If you notice that reticulum stuff, that endoplasmic reticulum, it does indeed that, doesn't it? Now, the way the artist drew this picture, you notice it's chopped open, right? So notice that the parts of the ER, and we'll talk in a second about rough and smooth. Notice like some of the other organelles there. Look, it's extending above. So it's a series of pipes that winds through the cytoplasm. Okay, I just wanted to make that observation there as we were talking about these. I know I kind of went off track about a plant and an animal cell, but let's do that comparing thing. So they got a lot of the organelles the same, right? That would be your compare. Let's do the contrast. Now, I'm going to kind of flip back and forth. Let's take a look at what's on the outside of this cell, something called a plasma or a cell membrane. Now, we'll talk about cell membranes in the next chapter, but to give you sort of a connection between where we've already been, I explained to you in the last chapter, there were these special lipid molecules known as phospholipids, right? You remember these? And you remember what I said, what happens to those when you put them in the water? Round up like the Indians. They make a hollow ball, don't they? Hollow sphere. And remember that time I said, wow, what a good thing to make a compartment out of. And then maybe what a good thing to combine a lot of, remember all those, that beautiful dance of biology that I always get worked up about. I said, you really make it work well, more efficiently, if it's in a smaller space. I think I used the analogy of looking for a penny in a, maybe it wasn't a penny in a phone booth versus a penny in a gymnasium, right? If you're in the phone booth, you're going to increase your chances. So this plasma membrane that we'll talk about more in the next chapter is made up of those phospholipids, right? It's a phospholipid bilayer. Okay, so take a look at that plasma membrane. Take a look at the cell. Take a look at, here we go again, the shape of that cell. If you were to describe the shape of that cell, what would you say? Oh, good. Thank you. Right to the point. It is sort of globular again, right? It's like that protein. Maybe you might have thinking, is it an oval? And if I were to tell you that just because the artist drew that cell this way doesn't mean that's the way they always look. Sometimes animal cells will, come on, pen, will look more round. Sometimes they're blue. They have all kinds of shapes to them, okay? They're not really a particular shape. I mean, they stay within constraints, but they don't. But let's flash over to the plant cell for a second. Now, take a look at that plant cell. Now, I understand that it has a complex type, what is that? Six-sided object that's surrounding that cell. And that's really only in this artist's picture again. But compare the shape of those two, or more importantly, contrast the shape of a plant cell and an animal cell. Who has a more regular kind of shape? If you want to see them in real life, let's take a look here at these. These are plant cells up here. Okay, we'll look at some of those under the microscope, hopefully. And you will look at some of these, hopefully. These are human cells down below. These are cheek cells off of the cheek of the inside of your mouth. And then LOD is a plant cell. You can see right away, if you look at the cheek cells, that, okay, some of them are round, but boy, this dude here, he ain't too round. He's much more this way, right? And then some of them are a little, this guy's got some crinkly hair. So, I mean, they can take on a variety. But these folks up here, relatively regular. As a matter of fact, that's where the name cell came from. The name cell came from a guy named Robert Hooke. Robert Hooke. Good old Robert. Huge name. Or huge nose. Sorry. Not huge name. I always wondered if his name and his nose were somehow together. Look him up online if you want to see a picture. But when he looked at cells through a microscope, when he looked at cork cells, a plant, he noticed that they were very regular. And to him, they reminded of the cells where monks live back then, very regular shaped room, same size, blah, 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 blah. So he thought they looked a lot more regular. Now, not all plant cells are quite that regular. You'll notice when hey, don't forget your stuff for the microscope tomorrow. If you're bringing something in, I'll check Cooper later for things. See if he's got anything. Anyway, so again, let's compare the shape between plant and animal cells, or contrast the shape. Which one is more regular? I think you guys already said it, right? You did say plant cells, didn't you? And the reason why it's more regular is something we'll talk about later on. Plant cells have something called a cell wall. See them down there? They have a cell wall. Now, that cell wall we have kind of talked about, because if you remember back in chapter 5, we talked about this thing. That was a carbohydrate. E. Right? So it's a carbohydrate, and of course I said we can't digest it. Your paper of your book is made of it. Metamucil. Right, Raymond, didn't you ask why you would do something like that? I was telling some friends of mine the other day, and it's from a young kid. Anyway, so there's plant cells have a cell wall. Now we'll talk as we kind of get into plants. Do we talk about cell walls again? Actually, no. I think this is about it. You know what they're made up of. Now let's go into function. Plants need a cell wall because it's something rigid. Animals don't need that. They usually have skeletons. They have different kinds of skeletons. Some are made of bone and some really aren't. Like worms don't have a bony skeleton. They have if you put your thumb over a hose, water's flowing out, right? You put your thumb over the hose, you know that pressure you feel behind the hose? It's called hydrostatic pressure. That's the kind of skeleton a worm has. Anyway, the point is, plants don't have any of that. So in order to stand up, in order for a tree to grow up against the force of gravity, it needs some kind of a supporting structure. Now don't get confused. You're saying, wait a minute, there's a skeleton. Yeah, only a name, right? The cell wall is really kind of the skeleton. The cell wall, which is a rigid, right? I held up a piece of paper on edge to show you the cellulose is rigid, stands up against the force of gravity. That's not needed in an animal cell, is it, right? Okay. So that's number one. What did we say? Cell wall in plant cells. Animal cells don't have a cell wall. Okay. Let's take a look. Don't worry about that. That's not one of the ones. I'll talk about it later. Let's take a look right here. Okay, a chloroplast. Notice that a chloroplast is green, not only by the artist's drawing, but if you'll look, there they are. These little dots. Look what color they are. Look at all those dots. Look what color they are. Get the pen out of there, Steve. There we go. Those little tiny things are called chloroplasts, and thank goodness they came about. We'll talk a little bit later on about where they may have come from, but without chloroplasts, well, we wouldn't be here talking about them, as a matter of fact. Chloroplasts were these important organelles that plants have, right? And you probably know what they do already. It's photosynthesis. We'll talk about them later on, but that's where plants are able to take the sun. Okay, let's just use magic right now. And by magic, turn it into food. Remember, I always said that's what I used to tell my kids. And I'm like, Daddy, how come lightning works? I'm like, oh, gosh, I'm trying to do the taxes. Lightning, it's magic, kids. My wife would say, don't tell them that. All right, anyway. So a chloroplast is in a plant cell. It does photosynthesis. Do you and I photosynthesize? Do us animals photosynthesize? We might want to. Some of us, maybe after being on a rocking ship after reticulating, might look this color, but we don't photosynthesize, do we? So difference number two, animal cells don't have a chloroplast, right? Don't see one there. And there's one right there. So we could use deductive, yes, we could use deductive reasoning. All things that photosynthesize have chloroplasts. Therefore, when I'm looking under a microscope at some little thing and I see a green dot, I'm like, oh, that's a chloroplast. It must photosynthesize. You guys remember early in the semester that I made some comment about chlorophyll and its association with photosynthesizing, and there was some, was it 2010, I think? A sci-fi movie where these scientists are analyzing the light that's coming off of a planet or even a star. You can do it. And remember, I told you that astronomers can actually tell kind of what is made up of that thing by looking at the light. And the dude looks up at the thing and he goes, chlorophyll. Of course, everybody in the audience goes, huh? And the nerds like me went, oh, chlorophyll. Yes, it was, right? So we started really big. All photosynthetic things have chloroplast. This thing I'm looking at has chloroplast. Ergo, prediction, small little thing. This one thing must be a photosynthesizer. All righty. Okay, so we got chloroplasts with different cell walls, a different last one you might not think about. Look at this thing right here, a central vacuole. Now, what is a vacuole? Well, a vacuole is kind of a broad term. A vacuole is kind of a sac. I think I spelled it wrong. Anyway, you can see how it's spelled there on the screen. It's a sac, and what do you put in a sac? Anything. Yeah, this particular vacuole that you see, the central vacuole, in a plant cell is typically a water vacuole. Just like Beth and or Lindsay said, it's a water vacuole. Notice that you'll see, but the reason I brought up vacuoles in general is we're going to see vacuoles later on that aren't water vacuoles. And sometimes the name of a vacuole is dependent on what's inside of it. There are pigment vacuoles, for example. If you brush up against poison ivy and you feel the irritation, that chemical that irritates your skin was stored in vacuoles. You touched it, they released that stuff on your skin. Don't touch me again. Weird, for some reason, I can rub up against poison ivy and have no effect. I don't know if it's some evolutionary advantage. All right, so plant cells have a central vacuole, typically a water vacuole. You look at animal, too far, go back. You look at animal cells, I see no such thing. Now, animal cells do have vacuoles, but they don't have a big central vacuole. As a matter of fact, in a plant cell, that central vacuole, this thing here, this open sac, can swell with, oftentimes it's water, so much that if you look at these Elodea cells, it's hard to think about it in 3-dimension, but the reason why those Elodea cells kind of look relatively clear, and then you see a blotch of dark green there, what you're looking at is you're looking through a really blown up balloon, okay, a square balloon, okay, but a blown up balloon. So the reason why it kind of looks clear with that dark is those chloroplasts. Matter of fact, most of the organelles in a plant cell are crammed up against the walls. In that picture, no, let me stay on this screen. If I could take one of those cells and we looked inside of it, this would be the central vacuole. That would take up that much of the volume. And all the other organelles would be crammed around the corner. So my question to you, I guess, since we're talking about the difference between plant and animal cells, why does a plant cell have something that to store water so necessarily? In other words, it needs to store water so badly that it grows and takes up such a big part of the cell. Whereas animal cells don't seem to be quite as concerned about water. There's no central vacuum. I'm not saying they don't need it, but there's not this concern to store lots of water. Why is that a big concern in plant cells and not in animals? Yeah. Where do plants get water? So an oak tree that's sitting in the middle of a drought that can see, they can't, they don't have eyes, but if it could, a river off in the distance. There's nothing they can do, right? Yeah, right, Raymond. There's nothing they can do. Even though that oak tree could, if it could, see the water, even though that oak tree could be within whatever a tree could walk. Anyway, you get my point. Animal could walk there, couldn't it? Animal, I'm thirsty. Get up, walk to the river, George. Oh, gosh. Remember Gary Larson? He used to write the Far Side cartoons? Probably don't. He always had one of the funniest cartoons. It was a deer that was born with a big bullseye on his side, just out of luck. And one deer says to him, wow, bummer of a birthmark, Hal. So from the plant point of view, bummer, you can't get over there to the water. Yeah, it can. But I mean, how far can the roots go, right? Typically, the roots of a plant, of a tree, extend out to about the distance that the leaves do, the crown, more or less. So even though, yeah. So, yeah, that's what they're doing. They're storing water. And since they don't know when the next time they get water, right? Beth and or Lindsay said it had to do with the weather. You're going to store as much water as you possibly can. Now, do the central vacuoles eventually shrink? Oh, yeah. Yeah, they do. Yeah, what typically happens in a plant cell, in one that's been dehydrated, excuse me, the central vacuole will shrink to the point that the cell membrane, they do have a membrane. Did they label it here? Yeah, there it is. They do have a membrane against, but they just have that additional cell wall. So that plasma membrane, because the cell wall will shrink, will actually pull away from the cell wall. And you look at the cell, and everything will be crammed in this little sac in the middle, and this will all be open space. Literally, the space between the cell wall and the membrane. That's a central vacuole. That's a dehydrated plant cell, boy. Yeah. When they spray, yeah, that's right, Tim. When they spray water, for reasons that we'll talk about later on, when they spray water onto produce, used to be when I was a kid, the guy'd have a hose. Now I think they kind of do it automatically, don't they? But when they spray water on there, what they're doing is getting that produce to plump up, getting that produce to take up a bunch of water, yeah, into the central vacuole. and push up against the wall. So when you grab that celery, it doesn't go back and forth. If I had a camera, you could see what means, but it's limpy. It's firm, it's crisp, okay? So what were the three things again? The shape, right? Plant cells have a much more rigid shape because they have a cell wall. That was one, right? One. Number two was the chloroplast, right? That means that you're a photosynthesizer. Plants are. There are no animals that are photosynthesizers. No animals. Sometimes polar bears will get a little green on them. Sometimes in the summertime, polar bears will have a little green tint to their fur. Have you ever seen that? I'll show you a picture, but yeah. Algae. It's a photosynthesizing plant for the most part. It grows on the fur. So they're not photosynthesizing, but the algae is. It's a relationship. Remember we talked about relationships between organisms, even predator-prey? Symbiotic, two organisms together. Okay, and the third difference was, we forgot, chloroplast, central vacuole, water vacuole. Got to have it. Remember, I'm a big proponent of understanding form and function. What's this thing do, or what's this thing look like, and what's it do? And how do that things compare together? How does the cell wall, right? Plant cells have them. There you go, form. How does that help the plant cell stand up? And why do animal cells need that? Form and function. So when you're studying, always put those two together. Okay, so you already saw this. We talked about the difference between the shape. Just do a couple of other ones. That's a cool picture up in the upper left. Those are stems. Stem cells are cells that haven't, well, the word is differentiate. They haven't ended up being what they're going to be yet. They're not yet a liver cell, a heart cell, a kidney cell, a gonad cell, a brain cell. They could be any cell. They're like before going to college, right? So stem cells, if you have brain cells that are dying off, like Alzheimer's and some of those other neurological diseases, if you could replace those brain cells with stem cells, you unfortunately can't replace brain cells, right? You don't replace brain cells. We talked about that the other day in class. Your cells every 7 years, right? Not your brain cells. Okay, and then down to the right, what's a skin gun? Skin gun. Down in the lower, while Tim's telling me a skin, the expression. Yeah. Okay, there you go. I didn't know there was such a thing. Cool. Yeah, it's exactly what they do. No. Now, I think there are certain brain cells that are Raymond, and I don't remember exactly, but I believe that, the ones that are really important for memory and those kinds of things, many of those are not replaceable. Or perhaps their replaceability. Remember, as you get older, entropy increases. So maybe what happens, and you'll have to check with me on this. Yeah, there's stem cells in the umbilical cord blood. But I think with brain cells, it's a matter that maybe that they don't necessarily are replaced. I think some of them aren't, but perhaps they're not replaced at a fast enough rate and they eventually start dying off, right? Sorry, I'm not more definitive on that. It's kind of, trying to remember from my days of A&P. But yeah, Beth and Lindsay, that's what's in the umbilical cord, is a lot of stem blood with stem cells. We talked the other day about surface area to volume ratio, why cells stay small. So I'm always curious in the exception. So I found the world's biggest cell, Valonia ventricosa. It's actually a single cell up to 5 centimeters in diameter. So the surface area to volume ratio certainly doesn't fit the pattern, but at least, for every rule out there in life, there are exceptions, aren't they? So I don't understand the physiology of Valonia, exactly how it deals with that surface area to volume ratio problem, but it's funny, it's not uncommon to find it. It's not a rare cell. You can look at aquariums, saltwater aquariums. My buddy had a bunch of this stuff. He had a name for it. I can't remember what he called it. But that's kind of cool. Where am I? What time is it? 8:36, 8:45, right? Okay, a few more minutes. Any questions on that? Yeah, okay, there you go, Raymond. Use inductive reasoning now and tell me how that cell makes a living. Or any of you. Yep, sure does. I mean, you can be green because you're Kermit the Frog, but you can be, if you're green, remember, inductive and deductive reasoning, isn't always perfect. Deductive reasoning. Surface area to volume ratio is the most crucial thing about cells. Therefore, cells should remain small. Valonia said, "Nuh-uh." Valonia said, "No, I can. For some reason that we don't need to get into here because I don't know. Number one, and number two, it's not really the scope of the class, but it's able to deal with that surface area to volume ratio problem. Okay, might have something to do with living in an aquatic environment, living in the water, right? All right. Any questions on that? Human cheek cells. I'm going to tell you in class look like blue fried eggs. They're blue because you stained them. You'll have to stain them. Now, you want to get that color of human cheek cells, too. We'll talk more about that in lab. You don't want to get them too dark, and I'll tell you how to control that. But let's move on, right? No questions? Okay, so difference between the animals and plant cells. Let's go to our first organelle. There's the nucleus. I'll talk about all those subparts here when we get to the nucleus page. Here's the nucleus. Okay, the nucleus is the control center, the top dog, the head cheese. The nucleus is the, well, like I said, it's what controls the activities of the cells, of the cell. And the reason why it does is inside the nucleus, you don't see it labeled here, something says chromatin. Here's chromatin. Chromatin is DNA. The little blue that you see there in the picture, that's DNA. So that's chromosome, right? And the chromatin, we'll talk more about this later in the semester, but the purple part is a protein. Purple part is a protein. The protein is called histones. More on histones later, but basically what you can see, DNA, chromosomes are kind of fragile. They're kind of weak little molecules. Remember, they're held together by just hydrogen bonds, across the bases. And you really don't kind of want your chromosomes getting messed up. So if you're not reading your chromosomes, if you're not using them, if you're not accessing the code, and you're not always accessing the code in your DNA, it's only when you need to make things, that chromosome is going to be wrapped up around those proteins like you see right there for protection. It's like putting glasses into those stupid styrofoam peanuts that are impossible if they blow all over your yard to find them. Right? It's kind of a packing material thing. And then when you need to read a piece of DNA, you unwind it. You read it, and then you wind it back up again. So that's what chromatin is. The word actually comes from the fact that we could stain it. When we first started looking at cells, chroma means color. Chromachrome. That song by, oh my God, Paul Simon. Anyway, years ago. Anyway, that's what chromatin is. We'll talk more about chromatin later. But you're the control center of the cell because you got that. You got the chromosomes. You got the code. If you know how to make things happen in a cell, if you know how to turn on that dance I've been talking about, that physiology, then ergo, you're the control center. Now, for some reason, that wasn't important in prokaryotes, but maybe because they're, again, no offense to you prokaryotes, I don't want to piss you off, you're kind of simple. All right, you don't have to really worry too much about chromosomes. That's not quite true, but good enough for us. Okay, the inside the nucleus, they don't put it here, does it? There's a liquid portion inside the nucleus too that these other things like the nucleolus and the chromosomes float in. It's called it's kind of like cytoplasm, but nucleoplasm. So that's that liquid filling in there. The nucleolus, not the nucleus, but the nucleolus right there in the middle. I'm just going to say basically one thing about that, or actually two things. I'm going to tell you what it does. The nucleolus creates this stuff. Shoot. There we go. Right? We talked about that the other day. That's called ribosomal RNA. Okay? And we're going to talk about ribosomes a little bit later on. We talked about those in prokaryotic cells, right? We mentioned that ribosomes is where proteins are made. It's the protein factory. Anyway, how you make that factory, how you make a ribosome is contained in that ribosomal RNA, and the nucleolus makes ribosomal RNA. So let's put that all together. Nucleolus makes ribosomal RNA, which makes ribosomes. And ribosomes, one more, you'll see what I'm writing in just a second. There we go. Nucleolus is responsible for creating ribosomal RNA, which is responsible for making ribosomes, which make proteins. So that's where the factory to make the proteins is constructed, or the information is located in the nucleolus. You don't often see a nucleolus. You might see a nucleus. You do, like in the blue-green egg thing, your cheek cells. That's what the yolk part is. That's the nucleus. Very colorful, isn't it, right? Chromatin. Okay, on the outside of the nucleus is really important, right? You don't want everything going in and out of the nucleus, because if you mess up the chromosomes, if you mess up the instructions, you might destroy the cell, right? So you're going to be very protective. So a nucleus has an envelope. It's got an inner membrane and an outer membrane. And by the way, any membrane in a cell, don't forget this, you can write this in your notes. Any membrane in a cell is made up of that phospholipid bilayer. Bilayer. Got to have two layers, because remember, it's one of them amphiphilic molecules. Any membrane. So a nuclear envelope has 4 layers of phospholipids, doesn't it? 2 sets of bilayers, an inner and an outer. Now, the cell membrane on the outside only has 1 layer, but man, I tell you, it's real important for the nucleus. We don't want just anything coming through, because if something goes through and messes with the DNA, it might be good. Remember, I said that's kind of like poking the devil with a short stick. Okay, when you mess with the DNA, you got to be real careful, because you're going to screw up the critter. You might make it better, all right? We won't talk about all this later on, but you're going to protect that. Things do have to go in and out, though. For example, ribosomal RNA, right? Ribosomal RNA has got to leave the nucleus and go out to where the ribosomes are. You may not know where they are right now, but that's what these little dots are. These are all little ribosomes. And we'll talk more about that later on. But that's the pore complex. There's a ribosome. A pore complex lets things in and out, not just ribosomal RNA, but very strictly controlled. It's like living in a gated community. Really cool pictures, right? It's an electron microscope. And then the lamina is kind of like that cellulose or fiberglass analogy. It's toughening, right? It's proteins that are laid down in a cross-hatch section to make very fabric-like. And that just means the nucleus is very tough. Okay? So the pore complex, what is a pore complex? If you look at it closer, it's a bunch of proteins around a hole. And what they do is those proteins basically swell or contract, and that means they open or close the hole. So if they swell together, and it closes the pore complex. They won't let them through. They contract, and it opens the pore complex. Okay, where am I? I know I got 1 more minute. I want to make sure I stop in a good place. All right, this is a good place. We'll stay on this slide, and then we'll finish here. If you'll notice right here, boy, when they're supposed to. I know that's not a good answer, Wyatt, but there's ways that the pore recognize what should go through and what shouldn't. And when I say recognize, again, it's fit. Oftentimes it has to do with fit. I know that probably wasn't a great answer, but right? It knows in some way that obviously has to do with molecules fitting together and stuff like that. What I circled up there, I want you to understand that that really complex membrane of the nucleus, that inner and outer, is confluent. In other words, it's connected to the ER, the endoplasmic reticulum. I'll talk about what rough is. It looks rough under a microscope, that's why. But notice it's connected together. That's important because there's this thing called the endomembrane system. Now, we'll talk more about that later on, but it's a membrane that's within the cell. And remember what I said, all membranes in a cell are made of the same thing, phospholipids and a bilayer. And then now, go back just one more step. Remember what I said, like dissolves like. Since the membranes are made of the same thing, this endomembrane system is a very good way to transport things in the cell. Right? But we'll stop right there, okay? I don't want to go any further. We'll talk more about that. We'll come back to it, all right? Okay, the exam tomorrow. I'll leave the recorder on, obviously. Anybody have any questions they need to get off their virtual chest right now that they just need to know about, and then anything specific? And then I can give you sort of an idea what the exam looks like. And I got a couple of sample questions maybe that I can throw out there so you can get an idea, especially if you've never had me for a class, which you haven't. You may not be familiar with the way I test. All right, anybody need to ask me anything before I get started? You know, it's an hour and 20, so you get the full hour. Take the full hour and 20. Remember I told you what, the last 15 minutes, right? Or did I say 20 minutes? You remember what I said at the beginning of the semester? 15 or 20 minutes, you can use your notes. It's a collection. There's some multiple choices. No, I did not. Didn't say 45. Good try, Tim. Yeah, it was. Okay, we'll talk about that. All right, I'll tell you what, we'll go with 20 minutes. Don't let me forget tomorrow. So you get last 20 minutes, you can use your notes. You can't use copies of my slides unless you've written on them. You can't just use pictures of my slides. Am I going to come around and check everybody's? No, I ain't got time for that. But if I walk by and you have a picture of my slides and you haven't made a bunch of notes all over it, if you haven't used it in class, that's why I want you to take notes. I'm not going to be happy with you, all right? Carbonyls and carboxyls. Let me get a blank. Let me get a blank thing here. There we go. Carbonyls and carboxyls. All right, functional groups. Yes, you should know functional groups for sure. You need to know them all. That's going to be a matching question, by the way. There's going to be a bunch of functional groups. You'll have to match them together. So back to Wyatt's question. Carbonyl. Let's start with where they're the same. Let's do a compare and contrast. Great way to learn. I'm sorry that my hand doesn't draw very well. But carbonyls and carboxyls both have this. Okay. They both have a double bonded oxygen. Here's the difference. A carbonyl ends in a hydrogen. That's why we call it a terminal hydrogen, whereas a carboxyl does that. It doesn't end in a hydroxyl. Remember, yes, that is a hydroxyl, but that's when it's by itself. It's not a hydroxyl, but a carboxyl or a carboxylic acid. Okay? Aldehyde. Generally a carbonyl. Remember, there are two types of carbonyls, right? There's also this one. Still got the double bonded oxygen, but it's somewhere in the middle of a molecule. Which one is this one? This one down here. Which one is this one? It's a carbonyl, yes, but I would probably want you to know more specifically what type of carbonyl. It's the only functional group we need to know a subtype, if you will. Why did I answer your question about the difference between a carbonyl and a carboxyl? Why folks are thinking about that? Cool. Anybody know what that carbonyl is that I'm pointing to with the question? That's an aldehyde up above because it ends in a hydrogen, has a terminal hydrogen. The one down below, though, you'd find it in the middle or in the midst, not necessarily in the middle of a molecule. There you go. Leave out the Y, but yep, I wouldn't. If you wrote that on the exam, for example, when it comes to spelling, since we've done it, I wouldn't take off for that. As long as you're close in the ballpark. I do want you to be as close as possible. Don't just give me some letters that you vomit on the page. But yeah, that'd be fine. Ketone, yes. Any other questions about functional groups? Good. Here's one for you. That's supposed to be a C. This molecule I'm drawing has two functional groups. Tell me which ones it has, and tell me what kind of molecule this is. And then tell me what type of a monomer it is. R means, rest of the molecule. Those aren't functional groups. Those the one on the left is. The carboxyl is, Raymond, but nitrogen is not a functional group. It contains nitrogen, but the name of it, there you go. Or amine, by the way. Oh, shoot. I thought I was typing. E or O works. Amino or amine. Okay, so it's got an amine or an amino, and it's got a carboxyl or a carboxylic acid. Remember, carboxyl also has another name. So this is the what? It's got an amine and a carboxylic acid. So what is it? Kind of an organic molecule, is it? Come on, I went over this in class the other day. Talked about it in the lab. I know we did. Beth and Lindsay. What kind of organic molecule is this? It has two functional groups. It's going to make those. Once you link these things together, it will. Yep. Amino acid. There we go. Thank you, Tim. Amino acid. So if I put a molecule on the exam, no matter what's in here, right? If you see these two functional groups, it's an amino acid, folks. Remember, this is the alpha. That's what that is. Carbon right there. Okay. One thing let me say about the exam. If on the exam you come up and there's a drawing or a question and you're just not getting it, call me over. I'm not going to give you the answer, but I'll be more than happy to kind of nudge you onto the track. And I'm not going to do that by hinting it to you, but I might tell you, no, think of it, there's another way you look at it. Like on this one, if you said, I have no idea, I'd say, remember, it's about the functional groups. If it has these certain functional groups, it is this thing. Okay. One of the other reasons, too, that I only give you the last 20 minutes to go over your notes is I found throughout the years that even though I've harped on students, is they'll bail out of the exam early. They'll take 30 minutes, whip through it real quick, get a 35 on it, and make a lot of dumb mistakes. So it's like, why are you hurrying? You got the hour and 20 minutes, You got plenty of time to go over your questions and make sure you didn't make a dumb mistake, to think about it again. Do some test mining. Y'all know what test mining is? Sometimes the answers are kind of tucked into maybe some multiple choice question. Like I might say, oh gosh, an atom with 2 electrons no, I'm sorry. An atom with 2 protons and a neutron, not including the electron, weighs how many AMUs? All right, so I got an atom. It has 2 protons. It's got a neutron. Sorry. This is what a symbol for a neutron. So how many AMUs does it weigh? Yeah, it weighs 3. Each proton weighs 1. Each neutron weighs 1. We're ignoring the electron. So yeah, it weighs 3 AMUs. All right, so, if you run into a problem like that on an exam, call me over and I'll be more than happy. Like I said, I'm not going to give you the answer, hint to it, but I will try to talk you off the edge if you're going down the wrong path. Sometimes it's just a matter of going, no, think of it the other way. Would you be able to recognize a saturated from an unsaturated fat, triglyceride? We went over that in class. That's something important because it helps us connect phospholipids, which we're getting ready to get into more deeply. And it makes sure that you understand that geometric isomers are bendy. Geometric isomers, yeah, there are 3 of them, remember? If you have a molecule like that, it's called a geometric isomer. And remember what that means is if I could turn that molecule on its side, if we looked at it flatly, it has a hinge. That double bond is a hinge. It can bend. The fact that it can bend, which is a chemical property, means that you can bend it and make things with it. The fact that iron is malleable, you can mash it into things, you can melt it and mold it into things, means we can make buildings. It's the same thing, folks. It seems like a little, just because it can bend, yeah, you can bend it. Just because those proteins attract, those side groups attract each other, yes, it bends. And bending means you can make shapes, and shapes means you can make structures, okay? What kind of a geometric isomer is this? In other words, is it a cis or a trans? CIS, but yeah, that's cis, right? How do you know it's cis, Tim? What tells you? What is the indicator that it's a cis version? The double bonds allows it to bend. That's right, Raymond. But the cis aspect that Tim picks up on is because if you look, the X parts, which there is no element X, but it's implied like in algebra, they're the same thing. So if that's a chlorine, they're both chlorines, are on the same side. If the X parts are on opposite sides, sorry for scribbling it all up, folks. Sorry, it actually would be over here. That would be the trans version. So there's a cis and a trans. Now they're two different molecules. Now they're both bendy, but they're two different molecules because of the fact that those X's are on opposite sides. Remember, diversity. Carbon's all about diversity. So back to my original question. Would you be able to recognize a triglyceride that is what I call saturated? If I could get a screenshot of that. Tell me something that you would need to know to tell me whether it's a saturated or unsaturated fat. If I put a triglyceride on the exam, if I drew out its chemical structure with all those carbons and all that stuff, would you be able to tell it's a triglyceride or not? And would you be able to tell if it was a saturated or unsaturated triglyceride? I'm trying to get an example to show you. Let's see if I can make this work. Can I copy that? No, it won't let me do that. There you go, right? So you're looking for double bonds. So you're looking down the carbon fatty acid tails. Oh, heck, I can just, let me just pull up the chapter. Makes it easier. Important to know because it's important for me that you guys understand chemical structure, and at least at this level. And the reason why that is because understanding chemical structure is going to help you later on understand why things are able to do what they do. So again, this is quite clearly not a saturated fat. And just for what Tim said, if you look at the double bonds, right? Not this double bond. Please don't make that mistake. It's not this double bond. That's a double bonded carbon and an oxygen. Isn't it? Isn't that a ketone group? Isn't that a ketone? Yes, it's a ketone. But notice, this is what Tim was talking about. It's got double bonds right there. See, that tells me that the hydrogens are missing. There are hydrogens that would be, let me erase my scribble. There are hydrogens that would normally be right here, but they're gone. It's not holding all the hydrogens it can. It's unsaturated. It's unsaturated, right? Unsaturated. Saturation refers to the hydrogens, and it's not holding all the hydrogens it can. And you are able to notice that. You can see that. I'm sorry if I'm kind of hitchy in my talking. I'm trying to do two things at once. But it is indeed, focus on that, right? Once again, associative learning. If I see a fat, and I look down the fatty acid tails, there are three of them because it's a tri, and I see double bonded carbons, I know it's unsaturated. If I see one, right, then it's monounsaturated, and if I see more than one, right, polyunsaturated. Okay, now, which ones are going to be liquid at room temperatures? Unsaturated or saturated? Don't worry about poly or mono, but which ones tends to be more liquid? Saturated or unsaturated? Okay, now think about what double bonds, here we go, form and function again, right? This is how you make sure that you don't make the mistake. Think about what double bonds do to atomic structure. Remember back in geometric isomers that we just talked about? What do double bonds, those double bonds that you see in the picture there in the second and third tails, means what can those tails do that the first one can't? They're bendy, right? They can bend. And when you can bend, that means you're more fluid, would you say? Bendy means fluidity, if that's a word. Fluidity, there we go. And there's something else missing in there, but you see how that works, right? So bendy is fluid. Now back to my question again. Think about that, what a double bond does. So which one of these fats, a saturated or unsaturated, is going to be more liquidy? maybe more fluidy, maybe more bendy. There you go, right? And now, sticking with unsaturated for a second, between mono and poly, let's say one that's really poly, which one is going to be more liquidy, the polyunsaturated or the monounsaturated? There you go. Makes sense? Right? Because remember, if they're all single bonds, then those tails aren't going to be very fluidy. Now, an example of a saturated fat, if you need a good visual, is butter. I showed you in class, right? Butter is a fat, but it ain't a liquid. You know, it can get a liquid here in Florida sitting outside. But yes, it's not a liquid at room temperature is what we say. But things like canola oil, vegetable oils, many of those are unsaturated, and that's why they're liquidy. That's why they're fluidy, okay? Which ones are better for you, by the way? Which fats are better for you? I always remember this because I know butter is not. Yeah. And the more poly, the better. All right, let me go back. I need a blank screen. Anything else you guys want me to? Let's see. There is a matching question. There's one on the functional groups. There's another one where Again, I'm looking at this question, and if you have any concerns about it or you're not sure what I'm indicating, it's a matching question where you have to match some images to some descriptions. Please call me over and talk to me. Like, for example, you might see something, and each exam might be a little different because I have several versions, which I just randomly pick a particular version, or the computer does actually. Oh, yeah, we did talk about it in Chapter 5. Make sure you know what ATP is, right? Make sure you know that it's adenosine triphosphate. Know that it's a nucleic acid, right? You should know that it's one, because I'm going to want you to know the different groups of organic molecules. I'm going to want you to know that there are carbohydrates. I'm going to want you to know their function. I'm going to want you to know lipids and their functions, definitely these. And of course, as I said, these two, the nucleic acids. Okay. While I'm on it, and I don't forget it, your essay question. I'm going to give you. Okay, you're going to have a question on inductive versus deductive, so make sure you really go over that. Here is the question. Let's see if it'll. Oh, it does. Excellent. That's the actual question you'll see on the essay, okay? I'm going to want you to know everything you can possibly know about proteins. And then you'll notice I didn't give you the whole question. And that's because I want you to do your due diligence. I want you to study. But your answer should include discussion regarding the following. And then I have 3, 4, 5, 6, 7 key points. And what I tell you about those key points is those are things I expect you to address in your answer. Those are sub-questions, if you will, or those are aspects of that answer that I want to be addressed. And again, in no less than 200 words. And I do have a rubric that I use that I grade this exam. And one of the categories is the number of words. So you'll lose points if you don't hit 200 words. 200 applicable words, appropriate words, words that aren't just stuff you repeat over and over again. I also have a grammar, punctuation, spelling part. As I said, I'm not big about spelling. I expect it to be close. But I do expect it to be grammatically clear. So the way I grade the rubric on that is if your grammar and spelling errors detract from the answer enough that I don't know what you're saying, you're going to lose points, or if there's just so many of them. So please don't give me text speech, right? Spell things out. Give me the commas and the periods and the capitalization. It's simply a matter of making it easier for me to understand your answer. Remember, when I'm reading your answer, when I'm grading it, I can't just simply call you up or email you and say, what did you mean when you said this? I don't have time for that. So make sure you explain your answer as if I don't know what you're talking about, as if your younger brother or sister or somebody came up to you and said exactly that. Hey, in 200 words, can you tell me about proteins? Now, if you think about the kind of things that should be included in your discussion, so the stuff after the following, you know I'm going to want to know about form. You know I'm going to want you to be able to tell me what makes up proteins. I may actually want you to be able to tell me what makes up those monomers that make up proteins. I'm going to want you to know terminology. One of the things I grade in the rubric, the third thing I grade, is using structural and functional terminology, using the right words. I want you to be able to say things like peptide bond, amine group, tertiary level. I want you to be able to tell me things and use words like denature. Those are important words. So make yourself a vocabulary list if you need, so that you understand what each of those words mean. Go through your notes. I've noticed years ago that students were horrible about taking notes, and that's why I allow this 20 minutes of note taking. But please don't abuse that and simply make copies of things. Make sure you go through your notes and you familiarized yourself with the information. I'm definitely, you can imagine I'm going to ask you about protein structure on this question. You can definitely imagine that regarding the following, one of those things down there is going to want you to tell me fully everything you understand about protein structure. Folks, if no bigger central element comes out of those first 5 chapters, to me anyway, it's protein structure. You've got to understand that to understand how everything else works. You have to understand how structure works in living things. You have to understand how function works. We're going to talk about enzymes, and they are proteins, and they're all about how proteins are put together. So please don't neglect that. Anything else? I'm willing to go on as long as you need me to. Last thing I ask in the rubric. I'm sorry. I have 4 levels. So length of the answer, your punctuation, grammar, sentence structure, you're using the right words, and finally I have a category that says you've demonstrated your overall competence. And these are worth anywhere, and I don't remember the points, but 0 to X amount of points. So the essay's worth 20 points, so it all adds up to 20. So it's 2-letter grades, so it's very important, obviously. But the last part is basically just kind of a summation. Did you convince me you understood what you were describing, or did you just simply vomit onto a page a bunch of facts? Please don't give me bullet points, for example. I want a written out answer, doggone it. I deserve it. I want a paragraph. I put a lot of effort into this class. You're going to put a lot of effort into this answer. I want it written out in such a way that it makes sense to me, that you convince me without a doubt this individual understands protein structure. And if you think, well, gosh, just for this class, if you're going on to A&P or medicine, it's all about protein structure in so many cases, folks. That's why I really harp on that. Raymond, tell me about guanine. What do you need to know about guanine? I'm sorry I went off on a soapbox there a little bit, but doggone it, I want you guys to learn how to write. And I know you know how to write. I want you guys to use that talent and that skill and that practice here in this class. Raymond, tell me, guanine, what you need to know. Did I lose you, Raymond, or is it just got a little bit of a lag? You guys can still hear me, right? If you all have other questions too as well, please go ahead. And we can always go over the mic as well. You don't have to stick here. Well, really, we didn't talk too much about function here too much, Raymond. They do have a function, but guanine is part of these 4, and I'm just going to put the abbreviations. Oh, man, type it in the box, Steve. Yeah, there you go. It was one of these 4. Adenine, thymine, I'm not yelling, I just didn't put the caps off. Guanine, as Raymond was asking about, and then cytosine. All right, and those are different types of nitrogenous, so they have nitrogen, nitrogenous bases. And nitrogenous bases were one of the 3 things that makes up a nucleotide. There it is, that structure and process terminology. These words need to be familiar to you. Nucleotide is a monomer of nucleic acid. And it's made up of 3 things. A sugar, either a deoxyribose or a ribose, a phosphate functional group, easy one to know because it's the only one that's got phosphorus, and one of these 4 nitrogenous bases. Right? Now, In DNA, okay, the function of guanine, and again, I'll get into more of this later on in the semester, but the function of guanine for our purposes is the sequence of letters in your DNA. That sequence of A's and T's and G's and C's, well, that's a code, okay? That's your genetic code. That, if you remember, is what I said, that's the recipe on how to make the protein. That's the connection we make between this code, which we'll talk a lot about at the end of the semester, and that primary level of protein structure, that sequence of amino acids. That's where the recipe comes from. So the function, at least at this level, back to Raymond's question, is it's part of the code. Now, chemically speaking, DNA is, like I said, an important and kind of fragile molecule. So the reason why DNA is double helixed, the reason why it's 2 strands, and the reason why you get adenine, hydrogen bonding to thymine only, and guanine always binds to cytosine, is because the reason why it's double helix is because it adds a little support. But think about the DNA as being a very important template. And with the double helix part of it, there's really only one part of the DNA that is the template of your code. The other part just matches up and kind of protects it and saves it. Right? I did it in class. I showed you the like the movie Gattaca, but I said if we got a T, T, A, G, C, A, T, A, not the Q, forget the Q, you should be able to tell me what's on the other side of the DNA. So not so much about function now, other than kind of hinting about the fact that it's code, but more function in terms of chemistry. It's the complementary of cytosine. So if you're working through that one right there, it's without the Q. That's the other side. Did that answer your question, Raymond? Okay, good deal. All right, anybody else? Yeah, I accidentally typed it in there. Oh, good. Yeah, scientific method. Yeah, scientific method. Definitely know that it's an observation, right? And you make a hypothesis, and you may make more than one, and then you experiment. Know what a control is. These are all reviews from the assignment. Oh, remember your assignment is due tomorrow too, by the way, the scientific method one. Okay, so know what a control is, be able to define it. A control, remember, is what we compare our results to. Yesterday in lab, the control was when we put the acid and base into water. Water has no buffering ability. Water is water. Gelatin or yeast or any of the other things we used, we determined had a buffering ability. They had the ability to absorb the change in pH, mostly the ones that were biological specimens, remember? Because they're most complex. But the control was when we did the into the distilled water, because there's no buffering capacity there. That's how we know the difference that occurred in the other things was because they were buffering it. All right? So make sure you know what a control is. Independent variable. The best way to, in your head, to remember those two is graphically. The independent variable is the X, and the dependent variable is the Y. And then word it. Is the air temperature dependent upon the month, or is the month dependent upon the air temperature? And say the words. Say, is the air temperature dependent upon the month? Yes, it is. Quite clearly it is. Right? The August is not dependent on the fact that it's 98 and 100% humidity and I'm sweating like a pig. Right? It's the other way around. So if you remember your scientific method assignment, and if you're going, oh my goodness, and you got to go back to it, remember, if you've already submitted it between now and 8 a.m. tomorrow, I'll open it back up again for you. If we look at the first set of chickens that were injected with the blood, okay, if we looked at that first set of chickens, what were the independent variables? And remember I said there are variables. So oftentimes students give me one, but don't give me the other. You got to have something to compare to. Don't forget the control. So what are the independent variables in that first round when they injected the chickens with blood? There you go. Which one was the control? Which was things normal, baseline, that we're going to compare it to? Yes, because chickens don't normally walk around getting injected with blood, right? That's the control. What was the dependent variable then? Notice Beth and Lindsay put 2 independent variables. They're correct. Both the control and the other one, the injected, we call the treatment, by the way. both the control and the treatment. Now, you might have more than two, you might have dozens, but anyway, those are independent, both of them, including the control. Which one is the dependent variable then? Yep, there you go. Whether they did or not. Getting the disease or not. There you go. So again, if you want to word it to yourself, if you're having troubles, if you go into the next one, which was what, the food, right? The different types of rice. Word it to yourself. Was the disease dependent upon the rice that they ate, or was the rice they ate dependent on whether they had the disease? So which one's the dependent? Do it in your head. Say it in your head. Does chickens getting sick depend on what they ate? Or is it what they ate depending on whether they were sick or not? Remember what's... Yeah, they got sick, right? So the dependent variable is whether they got sick or not. It doesn't change. We're still trying to figure out what makes the doggone chicken sick. As soon as we figure out what makes the chicken sick, we're assuming we'll figure out what makes the human sick. That's the whole point of the doggone experiment. Can't inject humans. Chickens don't mind. They don't fight back, right? So the dependency, again, is whether they get sick or not. The independent is we're saying, is it the food? Remember, that's the hypothesis. It went from the bacterium. That one didn't work. He rejected that. Is that a bad thing, by the way, that the bacteria of the injection with the blood, testing for a bacterium, that failed, that was it, obviously the not injecting chickens getting sick told them it wasn't a bacterium. Was that a bad thing? No. Why? Why was that not a bad thing? Right. Yes, what told them something, but why was the fact that their hypothesis that it was a bacterium? Yeah, there you go. Why was that not a bad thing? Because guess what? One part of the scientific method that we discussed is you go back and revise your hypothesis. Don't quit and say, the hell with it. Try something new. If you come in and you flip the switch, the light bulb doesn't come on and say, that's broke. Right? Change the bulb. Try something new. It eliminated the wrong. Yes, that's all part of science. Nobody says scientists get it right the first time out. They don't. Nobody does. Come on. Right? You go over and over and you keep trying and trying until you get it right. What's that saying that I love about sports? Amateurs do it till they get it right. Professionals do it till they don't get it wrong. That's what we're talking about here. You keep doing it and doing it and doing it. Keep revising that hypothesis. All right. Which one was the control of the rice, by the way? Let's see if you guys have been paying attention to what I'm saying about a control. Which one was the control? There were two types of the rice. Remember, those were the two independent variables. But which one was the control? Which was what, Tim? That's correct. You're right. It didn't cause them to get sick. The whole grain. That's right. They were always eating the whole. Remember, they switched over to the polished rice. Beth and Lindsay, back for just a second. I want to just kind of add to what you said about it eliminated the wrong thing, the blood. It wasn't the blood, folks. It wasn't the blood they were testing. The blood was the vehicle by which they delivered the thing they thought was causing. Remember, I asked you the first question, what was the initial hypothesis? I'm running over 9:20. If you need to go, go ahead. There you go. Thank you. If you need to go, that's fine. If you want to stick around and keep reviewing and talking about it, that's fine. It's up to you. I'm going to keep the recording going. You can always listen to it later. But if you are going, then I will see you tomorrow. Remember, 8 o'clock. And don't forget your Blackboard password. And don't forget to finish your scientific method assignment. So, all right. Yes, it was the bacteria was the original hypothesis. Remember what we did is we rejected it halfway through because doggone it, the ones that weren't injected, them doggone chickens got sick too. Please don't tell me about airborne viruses and maybe the ones that were sick gave it, because we never even tested that. Nobody's testing airborne stuff. We're injecting chickens, okay? Focus on one variable. So we injected them. They both got sick. Oh, gosh, that couldn't be it. Oh, maybe it's the food. Blah, blah, blah. Sure enough, that's what it was, right? It was the food. Do you remember what it was about the food? Hopefully you do. You just did the assignment the other day, I hope. Or at least soon. Or at least not that long ago. What was it about the polished rice? How come how come that gives you berry? Yep. Whole grain rice is better for you because the sheath on the outside has a lot of vitamin B. Thiamine. There you go. And a lack of thiamine causes beriberi. You have surprised how many students I've had throughout the years get to that part and say, well, you didn't tell us that. I'm like, you're sitting on the web. And I've told you before, I have no problem you checking the web for information. I'm like, yeah, do a little research. So yeah, it's thiamine. There you go. All right, what else we got, folks? I got office hours till 11:30. Now I am going to have to go step out for a second and go, use the boys' room here, but we can keep going. Under the assignments, Wyatt on the left and Blackboard, it says assignments, and then what does it say? Blackboard, I think. You have to scroll down. Yeah, Blackboard, which is beyond the spreadsheet of the assignments, and then there. And oh, they already opened up. The other concept maps for the next set of chapters is open up to energy and metabolism. I didn't change it to Wednesday, but you probably have no desire to do that right now, I don't think. Anyway, yeah, that's where it is. What else? No. You just submit it right online. You'll type in your answers. There's some multiple choice and kind of things you have to do, then you type in your answers. Oh, let me get my book. Oh man, do I? Yeah, I do. I keep the full book at home because I don't feel like lugging it around. It's heavier than an anchor. Okay, Wyatt. All right, let me see. 4-9. Oh yeah, know those. Be able to recognize them back and forth. Oh, be able to recognize what a functional group looks like. Like, okay. You all know what this is. I know. You know it's water. Why do you know that's not a functional group? And so many students will tell me that a lab practical. Water is a functional group. I'm like, what? How do you know that's not one, though? Yeah. By the way, speaking of 4-9, I'm not worried so much about knowing what functional groups do. That'll become, really kind of, if you understand that different functional groups and different things cause molecules to be more diverse and to fold and be different in different ways, that's great. But I'm not going to really specifically say what hydroxyl functional groups are alcohols, et cetera. But again, back to my question of water, how do you know that that's not a functional group? How do you know that water, if I drew it out like this, is a functional group? And how do you know that this is a functional group? I mean, other than saying, well, I know it's hydroxyl, so I know it is. But if you didn't know it was hydroxyl, but you did know something about functional groups, how would you know that is one? Too hard. Think of it this way. Let me draw the water again. Notice the bonds in water? They're all connected to something, aren't they? Right? But if we draw a functional group like that one I just drew a second ago, the OH, the hydroxyl, what's it connected to? It's connected to the rest of the molecule, right? It's connected to whatever alcohol or whatever molecule, or glucose or fructose. Remember I said it's the most common one you see on a molecule. It's connected to an R, a rest of the molecule. The reason how I know it's a functional group is because oftentimes the way it's written is we don't even bother to write the R. We say, you should know that's a functional group because that bond appears to connect to nothing. Because I'm focusing on this, not on the bond. I'm sorry, I didn't mean to go that far over the circle. Not on the bond itself. They're all bonded. This one is too. This one is too, right? So we say that functional groups have open-ended bonds. That's how we know it's a functional group. To differentiate it from a molecule. That makes sense? Sometimes, folks, I think what students do is they tend to make things more difficult than they need to be. So that's why I say, whoa, back off for just a second. Even in class, you'll hear me say that. Not a difficult question. Right? So if you find yourself doing that in the exam, overthinking things, again, call me over and I'll say, back off, back off, back off. Simple, All right. Phosphate functional group. Oh, yeah. How do we recognize that? For one thing, we're going to look for two things now. We're going to look for an open-ended bond. What else are we going to look for to make sure it's a phosphate?
Phosphate in the middle.
Yep. Listen, guys, I'm not going to screw with you and mix up functional groups. I'm not going to give you something like, like this and expect you to know that that's not a functional group. Now. That would be. That's a methyl. But I'm not going to do things like that. right? So I'm not gonna make it look like a phosphate with all the, remember phosphate, a lot of oxygens around it, right? I think 3 or 4 of them. I'm not gonna do that and put a nitrogen in the middle to mess with you. You're gonna see the functional groups as you see them. I try not to be tricky. I do expect you to pay attention to the questions. And so many times I look at students' answers and I'm like, they didn't read the question. I know they know this answer, but I'm not going to intentionally try to trick you. If on an exam you feel like, oh gosh, this is ambiguous, blah, 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 call me over, right? What else we got? What else we need to talk about? Do I need to talk about anything in chapter 1 and 2 other than the scientific method? I mean, come on, proton, well, that's 2, but characteristics of life, homeostasis, that's just a lot of jargon words you should know. There will be some multiple choice on the exam. How many did I put? I could look at it if that's, it's always funny to me, students, that's important, but if it is, that's fine. All right, so you know the essay that you got. How many multiple choice? 21. If you're asking me why is that, it's just the way the points add up. So there are 21 multiple choice questions coming out of a pool of several multiple, so you don't know exactly which ones you're going to get. But they're going to ask really simple types of questions. Let me give you an example of one. I'm not going to give you the answers. I'm just going to give you the question setup. Well, I will give you the answers. I won't give you the answer. So it says here, atoms with 8 electrons in their outer shells. Okay, so think about that for a second. Atoms with 8 electrons in their valence, I could wrote valence, or outer shells tend to, if you want to think about these, A, form ionic bonds. Now, here we can use our little emoticons. Do that. Give me a thumbs up or a thumbs down if you agree or disagree with that. Let's go A. So atoms with 8 electrons in their outer shells tend to form ionic bonds. Now, before you answer, remember in a multiple choice question, it's not always the right, but it's the most right. So if you're going, well, wait a minute, chlorine had 8 electrons in its outer shell. It became an ion and bond to sodium. Don't wait for just a minute. Metric conversions, yeah. Get it on your notes, write down kids have dropped over dead, that little conversion thing. I might ask you a few metric conversions. I don't think I did, though, actually, Wyatt. I think I saved that for lab. Okay, so atoms with 8 electrons in their outer shells form ionic bonds, form covalent bonds. That's B. C, are particularly stable and don't react. D, donate electrons. E, accept electrons. Or F, are very unstable. Now, I know I kind of went through those kind of quick. As I said, I'm not going to give you the answer, but would you be able to come up with the right answer? Atoms with 8 electrons in their outer shells, ionic, covalent, are pretty stable. Donate, accept electrons, are they unstable? Yeah, wow. You guys might be right or you might not. I don't know. Any questions on that, though? Oh, 8 electrons in their outer shells, Beth and Lindsay. Remember, when an atom has 8 electrons in its outer shell, that outer shell is complete. Ooh, good question to lead off from that. Oh, okay. Okay. Why, for the atoms we talked about, Raymond, we talked about this in class too, but for the atoms that we're discussing in biology, why physically, form and function, why is it that the outer shell only holds 8 electrons? What is it? Remember, shells don't exist. They're something we made up. But why is it that it only holds 8 electrons? Why doesn't it hold 10 or 6 or 2? Ah, there you go. Now you're getting it, right? Well, you may have gotten it before, I'm sorry, but orbitals make up shells. Orbitals are where electrons really exist. You might see a question on the exam. Electrons are in shells, electrons are in orbitals. You're going to go, oh, it's in orbitals. And again, that's an example of the best answer. They're not in shells, they're in orbitals. We collect them and make them shells, okay? Whew. That was so cool. You guys want another example? Let me give you another example. Let me find one that might tax you a little bit. Some of it's terminology. Oh no, I have to show you a picture of that. Oh, here's a good one. This is an easy one, but a good one. Most of the multiple choice questions tend to be relatively straightforward. Do you know the words? Do you know the basics? Okay, we have an element called xenon. Xenon has the atomic symbol Xe. Remember I told you there's no element X, but there is an element Xe. xenon. There, it's this atomic symbol. Xe has the atomic number 54. Okay, and it's the atomic mass 131. So 54 is the atomic number, and then the mass 131 AMUs. No J. Oh, got it. Big fingers. They don't have big fingers. All right. Let me ask some questions about the element xenon, see if you know what we're talking about here. How many protons in xenon? How many protons in xenon? Yeah, how do you know that, Tim? How do you know that, Raymond? How does anybody know that? That's what the atomic number tells you, right? Oh, wait a minute, Beth, for just a second. You jumped the gun. I was going to say, how many electrons in xenon? Tim and Raymond, you're absolutely correct. How many electrons in xenon? Go back to Beth and Lindsay's answer when they said just a second ago. It has to do with that. How many electrons, Tim? Yeah, how do you know that? Beth and Lindsay, how do you know that? Raymond, how do you know that? Balance. Positives have to equal negatives. Protons are positive. Electrons are negative. Atoms come out of the box, right? Created. Remember, we talked just briefly, atoms are created in exploding stars. But anyway, when that happens, they come out of the box in balance. It doesn't mean you're not reactive. It doesn't mean you have outer shells. You don't. Most of them don't. Only those ones on the far right of the periodic chart have full outer shells. But you come out with the same number of positives and minuses. So if you got 54 positives in xenon, whether you heard of that thing or not, you're going to have 54 negatives for the reason that Beth and Lindsay said a few moments ago. They balance, right? Positives equal negatives. Okay. Atomic mass is 131. How many neutrons does xenon have? Is that right? I'll do that in my head. Yes, that's correct. Tim, would you type in real quickly how you figured that out? Yeah, so you took the atomic mass minus the atomic number. No, not plus 77, equals 77. Yeah, there you go. Any questions on that? Sure, I get you. I do it all the time, right? Okay. Another type of xenon has the atomic mass of, let's say, 132. I don't know if that's true. I'm making that up. Doesn't matter. Let's say it does. So another type of xenon has the same. Another type of xenon, an isotope of xenon, would have how many protons? Protons. Please read the questions on my exams. An isotope of xenon would have, yes. What's it mean when something is an isotope, Raymond? Or anybody? Same. Same. One more word. No, what are these things called? What's carbon, hydrogen, nitrogen, oxygen? These things are called what? They are elements. Yes, there you go. Isotopes are the same element. It's still xenon, 54. Remember, the number of protons, atomic number of protons is what you are. If you have 55 protons, you ain't xenon no more. Okay? What if that isotope is radioactive? What do we call that? We have a special name for those types of isotopes. What do we call those? Okay. That would be correct, but sometimes we shorten the word. We just write radioisotopes, right? So if you see that word, yeah. Let's do a couple of pH things real quick. Recognizing numbers and what they mean. Solution A and B. We're going to compare two solutions. And let's say solution A has a pH of, 3. Solution B has a pH of, let's say 7. Okay, so A equals 3, 7. Bunch of questions I can get out of here. All right. Solution A, what type of solution is that with a pH of 3? As it pertains to pH, what type of solution is that? Okay. How do you know it's an acid, Raymond? What was the key that told you that? Okay, low numbers compared to what? What is the deciding, yeah, what's the deciding factor? For example, my next question would be, what kind of solution is solution B? It's neutral, right? So acids, neutral. Is that Spanish for it? Maybe it is. Acids are going to have pHs less than 7. Things that are 7 have a pH of 7. Okay. All right. Now, if you have a pH of 12, you're a what? Cool. Right. Very good. Now, let's go back to the original one. What we say 3 and 7, right? A is 3, B is 7. Isn't that what they were? I want to make sure so I don't confuse you guys. Okay. How much stronger of an acid is A than B? I mean, A is an acid and B is neutral, so A is an acid and B is not. But how much stronger of an acid is A than B? Okay, you got the right number. You got 4, but it's not 4 times stronger. Remember, this is what we call an exponential or a logarithmic scale. It's an absolute value, Tim, right? It's not going to be a plus or minus. But remember, this is a logarithmic scale, so it's a base 10 scale. So you're right. The difference between 7 and 3 is 4, but it's not 4 times stronger, not 0.4 times stronger. Anybody else want to help Tim out? Remember, it's a logarithmic scale. It's a base 10 scale. So 10 is involved. Remember, I showed you in class how to figure out pH by looking at that stuff. All right, so that's correct. However, this is what you do to figure out how much stronger it is. The number that you get, the difference between the 4 that we got in this example, that's the exponent. So it's 10. I can't make it, well, I can write it. 10, hold on. 10 to the 4th. All right, now how do we figure how much stronger of an acid? What is 10 to the 4th? What number is that? If you, yeah, there we go. Did you type out 10,000? Yeah, 10,000. Yeah, there we go. All right, so 7, 3, 12, acids, bases. Difference between here is 4. That's a 4 times stronger acid, right? No, it's a 10,000 times stronger acid. So here's an easy way to remember. If you want to know, take the 1, add as many zeros as the number tells you. 4. 1, 2, 3, 4. All right, 12. Solution C, we'll say. This is A, wasn't it? How much stronger of a base is this than B? Or we could say how much weaker of a base is B than C. Either way, how much? Is that 100,000 y'all are writing? Hope so. Yeah, it is 100,000. Yep, there we go. 12 minus 7 is 5. 10 to the 5th, 100,000. Perfect. Nice. So you see what I can do sometimes in questions is I can give you a setup like this, and then there's a bunch of different questions I can ask. Do you know what are acids and bases and what the numbers mean and relative concentrations or strengths, things like that, okay? Any other questions for me? I'm just kind of looking through here to see if there's anything that, I mean, everything else is pretty straightforward. If you've done your, as I say, if you've done your due diligence, you're studying, if you've done your reading, I don't think you have any problem with these essay questions. I really don't think you have any problem with the exam. My exams are complete. I do expect you to know a little bit about everything we talked about, okay? So make sure that you take your time, go through your notes. Don't just memorize things. If you need to make additional notes to yourself, do that. As I said, you can bring notes into class. You can bring notes you've written on that were the pictures of my slides. You just can't bring empty pictures of that. Because in some cases, there's nothing on there, right? Just a picture of a molecule. It's what I said that I want you to pay attention to, not the picture. All right, any other questions, folks? I'm kind of running out of things to talk about. If you have other questions, I'll be more than happy to. If you have anything else we need to discuss, but I'm sort of spent when it comes to that. But if you guys got something, let's do it. All right. No problem. Take care. You all have a nice day. Study hard. Cool. Thank you, Tim. I'll take a look at it. Click it right now before it ends up falling off the screen. I love it when you guys bring stuff like that to my attention. That's so cool. Oops. Stop. Okay, Wyatt. Take care. Bye, Raymond. You take it easy as well. Oh, you have to go right now, you're saying? Okay, I'm going to turn the recording off since we're going to talk about your assignment.